Welcome, everyone. Thank you for joining Southeast Regional Fruit and Vegetable Conference this year. I would like to welcome you all to this session. This is the afternoon session for vegetable production. I'm your moderator for this session. My name is Vivek Best. I'm the county agent in Dougherty County in Southwest Georgia. I work for UG Extension. And I would like to thank today's sponsor for this session, Farm to Market Consulting, for sponsoring the 2026 Vegetable Educational Conference. So without any further delay, I would like to first invite our sponsor for this session. They have a presentation ready, and once we go through this sponsor presentation, then we'll start with our first specialist speaker. Without any further delay, Ms. Christina? Okay, Ms. Christina, please, yes. Thank you.
I guess I can say hi for a second while I'm plugging things in. I'm Christina Monin. Nice to meet you guys. Got a quick video to share with you.
Our mission at Farm to Market is simple: to help growers take back control of their business. But let's talk honestly for a moment about what you're up against right now.
The reality is, the fresh produce industry has changed. The days where you could play the open market and hit big numbers, those $24, $30, even $40 markets, those days are fewer and farther between. What we're seeing now are extreme swings. It's either a $4 market or a $40 market, with very little middle ground. And unfortunately, we all know the truth. The $4 market shows up far more often than the $40. That kind of instability makes it nearly impossible to plan, to invest, or to grow sustainably. On top of that, costs keep going up. Labor, packaging, freight, fertilizer, insurance, everything. Yet the returns aren't rising to match those increases. And when you combine that with inconsistent support from marketers, you're often left carrying all the risk while someone else controls the sale. Many of you have experienced it. One week a marketer is pushing hard, the next week they're distracted with other crops or other priorities. Meanwhile, your product keeps growing and your margins keep shrinking. Another major shift is customer behavior. Retailers and food service companies have moved away from spot buying and toward contract programs. They want consistency, predictability, and long-term partnerships. But when you're not directly connected to those customers, you're missing opportunities to lock in stable returns and better plan your seasons. And we can't ignore what's happening with Mexico. Increased acreage, year-round production, and lower-cost inputs have created supply pressure that's dramatically affecting East Coast markets. For many growers, especially on the eastern side of the US, this has made traditional marketing models even more unpredictable.
This is why we created Farm to Market, to help growers shift from a model that reacts to the market to a model that builds the market.
We come in, learn your operation, understand your goals, and then we act as your in-house sales team, not a marketer, not a broker, not a middleman. We're part of your business. We help you establish direct relationships with retailers, food service companies, and distributors so you can stabilize returns, plan ahead, and eliminate unnecessary layers. And the best part, these customers belong to you. It's about giving you control over your pricing, control over who your product goes to, control over how your brand shows up in the marketplace, and most importantly, control over the future of your farm. Farmers work too hard to leave something this important up to chance, or to let someone else dictate the outcome. You deserve partners who are aligned with you, invested in your success, and committed to long-term growth. We want to build programs that last. Retailers and food service companies want consistency. They want reliability. They want growers who can partner with them year after year. And when we connect you directly to those channels, you're no longer at the mercy of the daily market. We appreciate every one of you and understand how vitally important you are. Our passion is helping farm businesses not just survive these changes, but thrive through them. We are Farm to Market.
Is this thing on? Yes. Hi, everybody. So thanks for listening to that. My name is Christina Monin, and my experience for the last 20 years, I have had the privilege of working directly for farms in sales and business development. My specialty and my expertise is developing long-term retail and food service business programs directly for farms that they keep. And So with this specialty, I've created I see a clear path where a lot of farmers are either working with mega marketers and rarely ever have direct business. And I see that the margins keep shrinking for the family farms, the farms that built America. And I feel very led, like this is my mission, to talk to you guys and say, hey, we can come in, we can analyze your business, and if you have a packing house, if you grow your own products and you're like, you know what, I'd like to stabilize my returns, I'd like to have some direct retail business, some food service business, come see us. We have a team that we actually would bring in and bring on the sales for your business. We are not middleman, so we would bring on the vendor directly for you for Walmart or Publix or whomever, depending on where you're located and what your commodity lines are. We'd have to analyze your business. But we would get vendor numbers directly for you, and then we would manage all the day-to-day sales for you, and that's business that you keep forever. So we have a booth 135 out on the trade show floor, but we also have a table right outside of here. And if any of this resonates with you, or if there's anything that we could do to kind of help stabilize your business and support the farms, I believe, not everybody is gifted in a lot of things. This is my one skill in life, and I'm very passionate about it. And if there's anything that me or my team can do to just at least talk to you, please come see us. I would be honored for the opportunity. Thank you.
Thank you, everyone. Let's give a big hand for Ms. Christina. Please join with me in inviting our next speaker. Dr. Cremonese is the extension entomologist in Auburn University, and I'm sure you all are excited to hear him out today.
Hi, hello everyone. I'm Paulo Cremonese. I work at Auburn University as extension specialist or IPM, vegetable and fruit pest management. And today I'm going to talk a little bit about the whitefly management updates that we have. I'm going to start now, and my colleague Dr. Andrea da Silva will finish up later with the remaining updates. But this is what we have been working in the state last year. So I'm going to start with talking a little bit about insecticide management. We investigated some sublethal effects of selected low-risk insecticides, more specifically 2 insecticides. We worked with NEC and Exirel, so growth regulator and a diamide. You'll see how it did affect performance, reproductive performance of the sweet potato whitefly. And this is part of the work conducted by my master's student, Ms. Priya Shai. Like we all know, I'm the first speaker today, but we are going to hear a little bit about whiteflies later. Sweet potato whiteflies are a major pest of agricultural, horticultural, and ornamental crops in the entire world. They are not strange to anyone. They are highly prolific. There's over 1,000 species of plants that they can feed on, they can survive, and they proliferate very well. They cause direct damage by phloem feeding, and indirect damage is transmitting disease, especially viruses, and also via honeydew, where sooty mold can be grown on the plants, and it will inhibit photosynthesis. And the management usually rely on chemical insecticides. Most commonly used are neonicotinoids, some pyrethroids as well, but we also have diamides and IGRs recommended for its control as well. So thinking about that, we thought about continuing the line of work of using sublethal insecticides, which are basically low lethal doses established for whiteflies, and see what it caused on the reproductive effects. And why did we wanted to check on sublethal effects? Well, sublethal effects is everything that doesn't kill the whitefly. It's an effect that's not directly linked to the mortality. So we can reduce the lifespan or delay the development, reduce fecundity, alter the host preference, modify feeding behaviors, or changes in virus transmissions. But today we only wanted to see what could be caused in terms of reproduction. So the hypothesis was that we selected an LC10 because we put these whiteflies very closely to the compounds, to the treated substrate. So that's why we use a very low sublethal concentration of both cyanotraniliprole and pyriproxfen, and that would reduce the net reproductive rate of the Mymisia tabaci compared to the untreated controls. So for this study, the experimental setup was very simple but very effective as well. We used cotton plants as the standard substrate. We selected only a single leaf, a single fully extended leaf, and we did the leaf tip treatment using the LC10 that we determined. And after 24 hours of that exposure, we collected whiteflies from the colony. The whiteflies are mainly grown on cotton. That's, like I said, the standard substrate. And then we release them under control conditions using this, using this arena here, this adapted arena that we have. And I'm going to talk a little bit more about this. But basically, the idea is that we have a very specific known exposed area that the whiteflies will feed on and lay the eggs. So they will be only allowed to feed, and that would be great. Look at that. Thank you, Ted. So let me keep on talking while we hook this up. Basically, they will have this specific area where they will be allowed to feed and reproduce. Yeah, it works. Right here, then we can basically compare the different treatments in the same arena. And after that, after 48 hours, we removed the adults. We counted the number of eggs, and we let them develop. After 10 days, we go after that, and we counted the number of nymphs. So the whole idea here is that in this arena, the whiteflies would be exposed to both treatment A and treatment B. And since they had the same area and they were released in a central point here, it would be a choice preference test for oviposition. So we did this in 3 phases. First, for adult whiteflies exposed to the LC10 of pyriproxfen against the control. The second phase was an LC10 of the diamide versus the control group. And then on the third phase, we compared both of the insecticides. These are some pictures of Priya setting up the experiment right here. As you can see, it was on controlled conditions. We made sure that we had this all standardized to reduce any possible errors or whiteflies choosing one substrate over the other. So it was all generalized. Light conditions were constant 24 hours. Whiteflies are fine with that. Over here is a close shot of what it looks like. And over here are the whiteflies that we collected from the colony. So jumping on the results for the LC10s that we use, the LC10s that we have used before with Georgia whitefly populations, while we all know that pyriproxfen disrupt immature development since it's a growth regulator, this LC10 did not affect the development of egg to nymph transition. It wasn't sufficient to produce any statistical results. So here are the number of eggs compared to the treatment, to the control. So the number of eggs in red and number of nymphs in blue, it didn't change. In our prior study, we have seen that the number of eggs was increased after treatment with pyriproxfen, actually. However, those eggs were not necessarily viable because, well, the pyriproxfen acts as a hormone that will help lay more eggs, actually, but they are unviable. Same thing happened here basically with cyanotraniliprole. So these are results for cyanotraniliprole compared with the control, and these are the results of both insecticides compared all together, although we seen here a little bit of variation on number of eggs for pyriproxfen treated under this LC10s that we study we didn't find any significant results. This showed us that, well, basically the next step is to reevaluate the sublethal concentrations specifically for our Alabama whiteflies, the whiteflies that we're using, since they have a different genetic characteristic probably. Jumping into the second experiment that we did, this one was not related to insecticide, but we wanted to check out greenhouse condition whiteflies under different levels of initial infestation in different varieties of tomatoes. So with this, we worked with Dr. De Silva's master student Nayli Cope and my student Priya Shahi that helped together, went up together to look at this. And basically, we wanted to study this on greenhouses. Greenhouses are everywhere in Alabama. They are controlled environments. Space and pests are commonly year-round. Right now in the winter, we have tomatoes being grown, so this is a very sweet home for the whiteflies to stay on. So for this, we wanted to test 3 different runs with different initial pressures of whiteflies, a low pressure, moderate, and high initial pressure. And we did counts standardized on the 3rd expanded tomato leaf. We used adults from the same colony, the same population that we had initially. These are the targets if you're wondering what we consider as low initial pressure or medium or high. Basically 1 to 2, 10, and over 20 adults per leaf initially. Greenhouse conditions are here, and it was a randomized block, complete block design. We did 4 reps for each in 5 cultivars, and the counts were taken weekly for 4 weeks. These were the varieties we tested, varieties usually used by the greenhouse company we were working with, Bonnie. Basically, we have Husky Cherry, beefsteak variety, a Better Boy, Roma, and a Sweet 100. So this is what happened when we had high initial infestation of whiteflies. Well, basically, whiteflies are present. There is no difference between each of the varieties at all, and they basically crash after the 4 week because it's too much. It's a very high pressure. Over here, we have an average of 60 whiteflies per leaves. So this is really a lot. So basically, under initial high infestations, there is no difference between any of the varieties we tested. And it's not very difficult to understand. This is the situation that we found there over the greenhouse. So whiteflies were all around. So it's amazing that they survive until the 3rd week, actually. Under moderate initial infestation, then we started to see a little bit more of a separation over here. Under the 3rd week, we had more whiteflies with the varieties Better Boy and Sweet 100 compared to beefsteaks, Roma, and Husky Cherry. But then again, at the 4th week, it was not significant anymore. But basically, we had a separation. However, when we had a low initial infestation, then the whiteflies start developing, and they choose more specifically to be more on Husky Cherry and Better Boy compared to Roma beefsteak and Sweet 100, which means that under conditions that we can manage whiteflies at low numbers, the varieties that we use might be a factor contributing into that. So in this case, these were the varieties that we had less whiteflies. Again, no insecticide management were used in this trial at all. So the take-home message right here is basically that the cultivar performance on the greenhouse depended on the initial sweet potato whitefly pressure. Under high pressure, we did not find any separation, just a uniform rise-fall trend. Under moderate pressure, we see brief differences only. Husky Cherry after 1 week, Better Boy after week 3, and they converge later on the last week. And under low pressure, we see a clear separation by week 3 to 4. Husky Cherry Better Boy had higher number of whiteflies. Roma had a mid number of whiteflies. The better cultivars under these situations were Beefsteak and Sweet 100. So host resistance trails are involved into this. So there are theoretically a lot of things getting involved with glandular trichomes, acyl sugars, ketones, terpenoids. We want to investigate that further, but they act over time. There is no knockdown effect like insecticides do. So this is going to be conducted under our investigation. For future directions, we want to do the metabolite profiling, investigate the settling, do a life table, use preference tests, and compare more different cultivars, or perhaps we can use different species of tomatoes as well. Under the long term, we want to extrapolate those results to open field testing and implement risk management systems for tomato IPM in Alabama. With that, again, I want to make sure that you guys have available the IPM resources we use. Again, check out the new 2026 crop handbook that was made available yesterday. Farming Basics app, still very interesting. The MyIPM vegetable for vegetables is a very good app. Growers can find a very good, reliable source there. With that, I want to acknowledge the team, Dr. Da Silva team, and the students. They did a very good job here. And with that, if you guys have any questions, I'll be happy to answer.
Any questions for the panel?
Yeah, have you done any work on predatory wasps for whiteflies?
Like in cars here. So we didn't, not yet, but that's definitely a possibility for the future. You know, like we can definitely implement that, especially transitioning into organic options on open field. But we can do that on a greenhouse scale. You know, that would be good as well, because many growers can rely on that as well. Yes.
In Alabama, when a growers have a whitefly infestation, do they CT1, CVT, or CV?
Could you repeat the question on the mic?
Yeah, can you repeat the question, please?
Yes. So when you have a whitefly infestation in tomatoes under field conditions in Alabama, do you see viruses?
Yes. This year we have seen some viruses in squash. I think it was CMV that we found on a grower consultation, but there's also TYLCV for tomatoes. But it's not like it wasn't a very big report this year. Whiteflies were not reported as bad this past year in Alabama.
Usually it's associated with those viruses.
Yeah,
right?
it is.
So why I'm saying that even a low infestation of whitefly will be able to transmit the virus. So
Yes.
whatever difference you see in the whitefly population or references may not be practical because there's few whiteflies will transmit the virus.
Yes.
So
Yes. Yeah, that's right. Because they are vector pests, it's very important to control them under low pressure. So as soon as we do the monitoring, it's important to go and intervene. Just because of that. It's
because
very important.
Asian sugar express lines we tested and also your the glandular trichomes lines we tested. Although they would reduce the whiteflies, but they would never able to reduce the virus transmission.
Yes.
So virus transmission will not able to curb.
Yeah. Yeah. And there's also the silverleaf disorder as well, which is associated with low numbers of whiteflies, regardless of the infestation, if it's high infestation or low.
Now, I was just wanted to see the reasoning behind those high, low, and medium pressure preferences. Because
Yeah.
even at the low pressure, you see the virus transmission. That will.
Yeah, well, when we talk about closed environment, you know, we have to take into accountability for the direct effects. Direct effects on their greenhouse is more important. You see a lot of whiteflies, and you start to see a lot of sooty mold growing on. It's
Okay.
yeah.
So these were not Bemisia. There were some others.
They are Bemisia.
They're Bemisia.
They are
but in the
Bemisia
greenhouse,
tabaci.
There
Yeah.
are different whitefly species in
There are greenhouse
greenhouse
whiteflies,
whiteflies, yeah.
but Bemisia tabaci is also very problematic under greenhouse conditions.
Okay.
In fact, they overtake
Just kind of follow up on what Babesh said. Do you actually see a lot of virus pressure in the greenhouse or not?
Oh, under greenhouse, yeah. I thought, yeah, I could have.
We have seen virus in the greenhouse. The problem is when we have the Bemisia tabaci affecting a greenhouse compared to the greenhouse whitefly, the Bemisia takes care of the greenhouse.
It does.
It's more aggressive
Very
compared
quickly.
to the greenhouse whitefly. And then when we have a high pressure, mostly in the border with Georgia, then when we have a lot of pressure in the field, usually you see the virus in the greenhouse. But it's much less common in the greenhouse than in the field. We
Yeah.
see much more virus in the
Okay.
field than in the greenhouse.
So then it makes sense to.
Yeah,
Most of our greenhouses are in Greenville. The Greenville is about
true. 2 hours.
80 miles from the border with Georgia and about 100 miles north from Florida. So it's in the southeast Alabama. So there is where we have we have about 80 greenhouses only growing like tomato transplants or vegetable transplants in general there. And that's where we have seen some of the
Yeah, it is. Yeah,
That's the company that we are working with that he showed
Yep.
the trials.
yeah, they have lots of issues, especially for herbs, you know. And it's herbs, you know, it's very important that you take care. You don't want a sooty mold because it's not only photosynthesis, it's the direct product over there. So this is due to the importance
That's
of
where
it.
they are winterizing right now. During the winter, they are reproducing and being kept into the greenhouse and mainly with birds.
Well, thank you very much.
Thank you all. Please join me again to thank Dr. Paolo and allow me to hand you the gift from the conference.
Thank you very much.
Thank you very much. I really want to go through these presentations really quick, so let me invite our next speaker for this session. We have Dr. Bhavesh Datta, who is the professor and extension plant pathologist at the University of Georgia. He is going to present the overview of work done by multiple scientists over more than 5 years, compiled into an overview and summarized into a risk assessment phone app to be further developed by ongoing researchers. Please join me to invite Dr. Datta for his talk.
Thank you, Vivek. All right. Before I start, this will be a team talk, so I'll be teaming with Dr. Anup Malik, who is also my postdoc, and he'll be talking about the host resistance in snap bean against Cedar golden mosaic virus. So the title of my talk is Risk Index and Host Resistance, two different things. Okay. In first half, I'll talk about more of applied, highly translational work. And the second half, it's going to be more genetics and how we developed or how we identified resistance in snap bean against whitefly transmitted viruses. So if you remember, I've worked with Babu and a number of horticulturists like Dr. Coulong, Dr. Andre De Silva, now Ted McAvoy, and a number of scientists from Florida, South Carolina, and so forth. So this is the outcome of those collaborations. So risk index. I'm hearing the voice from other room too. What you're seeing here is the squash impacted by viral complex, whitefly transmitted viral complex here. This is squash, and look at the amount of whiteflies over here, and this is snap bean. So in 2015 and 2016, when we started this project, we had continuous field failures, especially in fall-grown squash in Georgia, but particularly in southern Georgia. In 2016, we had close to 60% crop loss in yellow squash and also in other cucurbit, but yellow squash was hard hit. And the following year, subsequently in 2017 and 2018, we have close to 70 to 80% crop loss in snap beans. The entire snap bean industry in the fall in Georgia was decimated by this virus. So my talk will be first on squash, then on snap beans. With squash, I'd like to acknowledge two funding sources. So the first funding source is by USDA SRI project in 2018, funded in 2018 to Dr. Scott Atkins in Florida. And also subsequently in 2019 onwards, we are lucky to get a continuous support from USDA UGA Cooperative Agreement led by Dr. Alvin Simmons. Okay, so first half of the talk, I'm going to talk about how we developed a risk index for our Georgia squash growers, our southern squash grower, to predict risk of whitefly transmitted viruses in squash for fall. So this particular work was a part of a PhD dissertation of my previous grad student, Dr. Clarence Goddard, and he did a fantastic work on this thing. So there are a number of people been involved. So like Dr. Da Silva, Dr. Canadian, Dr. Sparks, Dr. Babu, Dr. Camerite, and myself. So risk index, you all, those who work on peanut, or those who have been involved with peanut, they know this peanut RX. It is highly influential, highly impactful, and well-recognized peanut TSW predictive model. Okay, here are RX. Here, the different variables being used, like peanut variety, planting date, plant population, at plant insecticide application, row pattern, blah, blah, blah. So number of variables are being used, and based on the use of these variables, they predict the TSWB incidence in peanut. That was the encouragement for us, that can we look at some of the variables our squash growers or cucumber growers been using to predict what is the risk of the whitefly transmitted virus incidence in the fall. So we looked at a number of things, a lot of different things we have looked. But these are the 5 important variables which we came along and to know that these variables might be important. So one of the variables is use of row covers. Another is planting date, mulch type of squash you are growing, either winter squash or summer squash, or planting material, either seedling or seeds. Based on that and based on the relative importance of each, we have devised a model and predicted the model and later validated it. I'm not going to show each and every trial. So for each variable, we did at least 7 to 8 different trials across 3 to 4 years. Okay. So I'm just going to give you a snapshot of one of the trials we did, where we combined use of UV reflective mulch. Instead of white, we have a UV reflective mulch. Row covers, look at the floating row covers, and planting date together. So what you are seeing here is the outcome of these practices, or the combinations, at harvest maturity. Look at in the fall, when we grew squash under very high whitefly pressure without row cover or any plastic mulch or any reflective plastic mulch, look at the amount of silvering you get. Now in contrast, look at this one. This here, squash were grown on reflective UV mulch, and then we also had a row cover for just 10 days after transplanting. Look at the difference. There is no or very, very less silvering, and when we looked at the disease rating, there were very less virus incidence here. In comparison, we can also look at when squash were grown on just UV mulch, but no row covers. Despite we use UV mulch, but without row covers, we have a good bit of silvering and also a good bit of virus incidence. Alternatively, we also looked at white mulch, white plastic mulch where squash were grown, but only with row covers. So here you can see there is some silvering, but there are a lot of green tissue and less virus incidence. More like later we found the row covers are the drivers of reducing the pest as well as virus incidence rather than this silver plastic mulch. So based on all the permutations and combinations and a number of different trials, we came up with different risk index. Okay. For each predictive variable, we ranked the variables, and based on their importance to disease incidence, we gave them the pointers. So for example, planting date, use of row cover type, squash, planting material, and mulch type. Look at this one. For planting date, if we plant it before August 11th, it has a risk point, less risk point. But if you plant between August 26th to September 20th, probably you're at a higher risk. Row cover, if you use a row cover, you are at a low risk. If you do not use a row cover, you're at a higher risk. Squash type, if you use a zucchini and all, you have a low risk. If you're using yellow squash, it has a higher risk. Planting material, if you use seed, you are at a higher risk. If you are seedling, you are at a lower risk. And mulch type, if you use a white mulch or UV mulch, more or less the UV mulch had a lesser risk, white mulch a more risk. So combination of these gave us a total risk range of 45 to 75 points, moderate risk is 80 to 115 points, and high risk to 120 to 150 points. Okay. These numbers were not randomly assigned, they're all based on statistics. Okay. I'm not going to talk about those, but these are the risks. Now, this is all about the model and the risk index. Now we have to have a bigger job at hand is to validate it. So first 2 years of validation was done at UGS station in the fall, where we chose some of these variables in a combination that will make it as a high risk, moderate risk, or low risk. So at the end of the season, this is how the high risk looked like, this is how moderate risk, and this is how low risk. So we also validated our model in close to 22 different commercial fields. So what we did, for 2 years, we went to squash growers. We asked them what are the different management practices they're doing. And based on that, we wrote what are their risks. And then we gave them that this is probably you're at a high risk, moderate risk, or low risk. And they said, okay. Then at the end of the season, we came back and we rated for the virus incidence, not the insect, virus incidence. And look at this thing. It was a highly significant with a decent R value. It is highly correlated. It means the risk which we predicted is what we found, what we validated in growers field. So this is one of the examples of a grower field. We told him that you are at a high risk. He was growing on a white plastic on August 25th, transplanted, no row covers. So he was at a very higher risk because his risk was 145, and indeed he had a 40% yield reduction. This is an example I'm showing you. So this shows our model and risk index is validated and is working. So in summary, we used random forest analysis and random forest to rank these variables and make this risk index, and we also validated it. So we went forward and we developed an app for our growers. It's called Squash RX. This Squash RX, it's free, available free in both Apple and also in Android platform, and you can use it. Here, if you open the app, it will ask you which type you're growing, either yellow or zucchini. If you say, let's say yellow. Next, what's your material, seed or seedling? More likely seedling in Georgia. Next, are you using mulch or no mulch? If more likely you're going to use a mulch. If you're using plastic mulch or UV mulch, you can choose. Are you using row cover or no row cover? You can choose. Then what's your planting date? I just chose a planting date of 30th of January. So then these are the summary. You choose, and then it calculates based on the risk equation we got. And it gave you a low risk. Because you're planting in winter, so it's a low risk. But if the same thing would have done in August or September, probably you would have moderate or high risk. But anyway, it's a free app for everyone. Now, good thing is that a lot of our growers have downloaded it and they have started using it. I got some of the feedback from the growers that it's actually helping them with this one. Luckily or unluckily, we had a lower risk, a moderate level of infestation this year. So we were good. But any years which we have a high infestation in summer or early fall, probably this app will help them to make the decision in either choosing a planting date or modifying some of their culture practices based on the variables. So with this, I'd like to invite my postdoc, Dr. Anup Malik. He'll talk about snap bean and host resistance. So.
We'll do the Q&A at the end.
Yes.
Yes. Hi, good afternoon all. I'll be talking about SnapBean from now. So I would like to say something before I start my presentation. So what is the last time when you see your SnapBean fields are infested with whiteflies, despite of spraying, following all the recommendations? So I think this is the frustration when you do everything and this comes up with this project to work on SnapBean. So we wanted to find some SnapBean lines which can protect themselves. Those have those genetic resistance in them in the field. And with these slides, I would like to show how close we are towards our goal. So let's move to the first slide. So on the left, you can see there is a healthy SnapBean plant. And on the right, here you can see there is a plant which is completely infested with Cedar Gold mosaic virus, which is one of the virus transmitted through whiteflies. So there was 80% crop loss, which was observed in 2017. Moving to the next slide, we did some phenotyping in the field. So here you can see there are some lines over here which are completely resistant, and they show a very good growth. On the other hand, there are some lines over here which are susceptible. So we did this study, and we did the disease severity, and you can see that we have a AWPC panel. So we calculated the AWPC on a scale of 0 to 20, and 20 to 50, 50 to 65, and 65 onwards, and we classified these scales on highly resistant, moderately resistant, susceptible, and highly susceptible. So we have a panel of 373 lines in SnapBean, which we tested, which we phenotyped actually. And on the right, you can see a graph of disease severity and AUDPC of these 373 SnapBean lines. Moving to the next slide, so we started this phenotyping in the field from 2018 till 2023. So these 6 years, we screened several SnapBean lines, and in those lines, you can see over here, like in 2018, we have 82, 2019, 74, 2024, 86. So these are not the lines which are just screened once. These are some of the lines which are minimum screened at least twice in the field. Some of the lines are even screened thrice in those 6 years. So these are not the individual lines. So based on the screening, we classified these lines as I said, highly resistant, moderately resistant, susceptible, and highly susceptible. So in the field, when you see some lines which are not infected, some are infected, that's a question comes in about DNA. So we did some genome sequencing of those lines, and what we found, there is some error in the font here. So we found some 32 million raw SNPs. So SNPs are just the small modifications in the genome of the SnapBean lines, which actually make them either resistant or susceptible. So we found some SNPs in the SnapBean lines which are 32 million, and these are actually 65K filtered SNPs which are not because of any error, and these are uniformly distributed on 11 chromosomes of SnapBean. So here you can see the graph and the SNP density. Moving to the next slide, so these 65K SNPs which I am talking about, they are actually divided into 3 clusters. So you can see we did a principal component analysis, and we found that there are 3 distinct clusters we found, and these are all 373 SnapBean lines. Moving to next, so once we get the SNPs and we know they are present in all the chromosomes, so moving to next, we did the annotation, which is called which SNP is actually linked with which of the gene in the genome of SnapBean. So we did some SNPs, and we found that 90% of the SNPs, I call SNPs as SNPs, so 90% of the SNPs are from intergenic variant. There are few from upstream gene variant, there are few from downstream gene variant, but you can see over here the most are of the SNPs are from intergenic variant. So out of those which are over here, we found 2 of these SNPs which are highly associated with our phenotype data. and we found one SNP is on chromosome 10 and other is on chromosome 6. And they are one from 5' UTR region and one from intron variant. So moving to the first SNP, it is actually associated with a gene called mitogen activated protein kinase 1. Moving to the next slide, there are a lot of, because this is a family of MAPK genes, and there are a lot of roles in rice and other plants of MAPK cascade, which shows that they have a very prominent role in immune responses. And in rice, it activates immune responses upon the perception of chitin, and it also uses a signal transduction in a lot of plant immune responses. So the gene which is associated to SNAP-bean, which we got, is actually studied before in a lot of studies like Wang et al. And Sun and Zhang. This gene also has certain roles in viral protein MAPK pathway, which actually downregulates, and it helped the infection caused by DNA and RNA viruses. So we are still in the process of study that what is the role played by our gene in SNAP-bean towards the resistance. The next SNP which we got is from chromosome 10, and it is from a K-box domain protein family. And there was a study in 2024 by OK et al. And it says that some of these genes are involved in drought stress. So the infected lines are also going through a stress. So this might be another gene which is associated towards the resistance of SNAP-bean. So we are still in process of doing more validations in the lab by using CASP markers and other validation processes to find out like whether the two SNPs which you got, they actually play a role in resistance or not. With that, I'll just give you a brief, a quick recap, I should say. So we did some SNP density on our genome. We did some LD plot analysis, which shows how these SNPs have their linkage disequilibrium. We did a linkage disequilibrium study per chromosome. We also find out how many clusters these 373 lines are divided in. And we did some population structure studies to find out in each line how much of the genome percentage is there. We did some phylogenetic study to again find out the cluster and kinship study. We also did some, as I said, we did Manhattan plot to see the association of the SNPs to the phenotype data, and eventually the SNP annotation. With this, I think I will hand over the presentation to Dr. Datta again.
Last one, couple of slides where we finish. So where are we going from here? We also been collaborating with Syngenta and their advanced lines. So some of the advanced lines which we identified will be also included in the Syngenta lines. Some of the Syngenta lines we have identified to have resistance will be included. Plus, we also have PI lines identified by our group will be included in UGA SNAP-bean breeding program by Dr. Amol Nankar, who is our UGA vegetable breeder. He'll be including it. And some of the stakeholder-oriented impact, Squash RX is already here, being downloaded by our growers. Silver mulch is being used in Decatur County, which borders Georgia and Florida against and Alabama too. Again for tomatoes. In squash, use of transplants is now a preferred practice rather than a seed. Growers are aware of the risk of Sida golden mosaic virus because it's a reservoir, Sida as a reservoir, and we're going to have a good choices of varieties very soon. New highly resistant lines were identified and will be incorporated in the UGBD program. So these are some of the impacts of our work. And with this, I'd like to have questions. I don't know if I have any time.
We don't have a ton of time, but we can do just one or two questions. If you all have any.
Yes.
So it's a virus complex, right? So what are the viruses we know that we should know?
Good. So you're talking about squash or on SNAP-bean?
Either one.
Okay. Let's start with squash. Squash is a complex. So we have a cucumber leaf crumple virus. We have your CCYB and CALRDV. the 3 viruses. And in SnapBean, we see Cedar golden mosaic virus. It's a driver of the infection, but sometimes it is also mixed with cucumber leaf crumple virus. But in most cases, it's the Cedar golden mosaic virus that drives infection. Any other questions? If not, thank you all.
Thank you very much, Dr. Dutt. And I don't have a token for you, token of love and appreciation for you, because you already got earlier this morning or yesterday. For the specialist who had multiple presentations during this conference received the appreciation at their first presentation. All right. Thank you again, Dr. Datta and Dr. Malik. I'll go with the next presenter today, and please join me in inviting Dr. Ted McAvoy. He is assistant professor and extensive vegetable production specialist in University of Georgia at Tifton campus.
Thank you, Vivek. So today I'm going to talk about host resistance in different cucurbit crops and species, as well as tomatoes, both commercial lines and experimental. And so before I start, this is the work being done by my graduate student Nirmala, and this is an extension pub that we have available if you'd like to look at it. And also, Nirmala is Vivek's husband, or wife. That's right. So just based on our last couple years, as was mentioned, whitefly feeding causes silvering, so we do have differences in silvering symptoms between crop species of cucurbits. These are the viruses that are transmitted by whitefly. We were talking about the virus complex: crumple leaf virus, cucurbit chlorotic yellows virus, cucurbit yellow stunting disorder virus. The DNA crumple leaf attacks the growing point, and it's all crumpled up and twisted, whereas the other two are yellowing viruses and attack the older growth. So we kind of just made a guide with short season crops and long season crops. A lot of it's based on what we've seen, but also in our research plots. So out of the, all these crops are widely grown in Georgia in the spring and the fall. We have problems in the fall where whitefly builds on cotton, and then it comes into our, getting virus from weeds, and then we get the virus in our crops. So cucumber doesn't silver. It doesn't show symptoms for cucurbit leaf crumple virus, and it's low to moderate for the yellowing viruses. So out of the short season crops, cucumber is what we'd recommend. Now we did, in the past I've shown varietal differences for yields. I think that's mainly just difference to how the variety performs, not necessarily virus differences. As mentioned in the Squash RX, zucchini is, although same species, zucchini is better than yellow squash. Yellow squash is the worst. So if you have the opportunity to grow zucchini rather than yellow squash, that'd be preferable. But we do have differences in varieties as well. And this is just still from that extension publication. You could see, the cucumber doesn't silver and it doesn't crumple, but you get yellowing at the end. But all the fruit's marketable. The fruit doesn't become unmarketable due to virus. Zucchini, we have little silvering, not as much as yellow. Yellow is just the worst with crumple, and so that's why it's bad. And in the end, zucchini, most of the time, the fruit is still marketable, maybe lower yields, but on squash, it really affects the marketability. You get green streaks and spots and malformed fruit. All right, now with long season, this is an opportunity for people to grow different crops. The other crops I mentioned are widely grown. We don't grow too much butternut or calabasa, but they're cucurbit machata, and they had much better resistance relative compared to other crops. Definitely don't recommend acorn, Hubbard, pumpkin, or watermelon. They did terrible. I would say, to give the breeder a plug, Cecilia McGregor has been working with different species, and the watermelon coming out of that program looks excellent as far as virus resistant. Now we got to work on size and quality of that fruit. So just was the butternut that I was telling you about, relatively low silvering, does good on crumple, and has marketable fruit. Ceres and Genesis were the two varieties that did well for us. So that's all stuff that's been done in the past. The way that we set up our experiments before, we blocked by crop, so we couldn't compare head-to-head crops. But this time we put all the summer squash, zucchini, and yellow squash together so we can compare them. And this is what we got. We also included known, our best performers for the past and susceptible from the past, and we got some new varieties. Some of them are breeder material from H.M. Klaus as well. So here is our marketable fruit, Renegade. That's a zucchini. That's been doing excellent for us for the past 3 years. Rocchio, that's a gray zucchini. That's been doing excellent for us the last few years. One of the H.M. Klaus varieties, and then this is probably the industry standard for zucchini's respect. Some did very poorly. Gold Star and Gentry have historically done very poorly, but also Lioness, which has done well in the past, really did poorly this year. So, that's one to watch. That was surprising. And this is the area under the disease progress curve, so disease over time as it accumulates. What's very nice to see here is that the H.M. Klaus varieties have really low symptoms of cucurbit leaf crumple virus. Also, as I was talking about, the crop from here this way is zucchini, and here this way is summer squash. So you see vast differences. This is yellowing symptomology. Again, maybe a virologist wouldn't love it, but I was just looking at symptoms in the field and not testing for the virus, but just looking at yellow on the lower leaves. Again, the H.M. Klaus varieties did excellent, as well did Respect and Renegade. So I think there's a lot of good options and choices, and hopefully you can continue growing your crops, making better decisions. Now moving over to tomato, and I'm going to have Vivek hand out a questionnaire. I would really appreciate if you fill it out. It's just yes or no questions, super easy, front and back. And all I want to know is, did you learn anything? With the Georgia Department of Ag grant I got, last year I reported that 50 people saw my presentation and learned something. They said, well, just because they attended doesn't mean they learned something. You have to ask them if they already knew it. So please fill that out. So with tomatoes, we have one of the whitefly transmitted viruses, tomato yellow leaf curl virus. That's been a problem in the tomato industry since the 90s. It's standard in the fall to have resistant varieties. Everyone's growing resistant varieties. But what they've been seeing for about the past 4 or 5 years is a breakdown in resistance. So ones that used to be highly resistant still are better than susceptible, but they're showing more symptoms and not performing as well as they did in the past. So we wanted to test some more varieties. You can see the yellowing in the top of the plant. But something that we discovered a few years ago, working with Sudeep Bag, the virologist at University of Georgia, is tomato chlorosis virus. Again, it looks like a nutritional disorder. You get yellowing on the lower leaves. You could get cupping and brittle leaves. I've seen it before. I wouldn't have thought it was a virus. If you're growing tomatillo and in the fall, it'll be completely yellow, look like you didn't put any nutrients on it, or worse, it's this virus. The literature has suggested out of Spain that mixed infection with tomato chlorosis virus and tomato yellow leaf curl virus is causing a breakdown in resistance, especially to TY1. There's 6 resistance genes that are currently incorporated, TY1 through TY6. Most of the commercial varieties have a stacked gene of TY3 and TY6 that we're currently using. In addition to that, the virologist Sudeep Bag has been collecting virus samples. And so what they've done is they sequenced the TYLCV that we have in Georgia and compare it to the Israel isolate, which is the original isolate that these were developed for resistance to. And this data has been published. It differed significantly enough that they've recharacterized it as TYLCV GA, Georgia. So we have a modified virus and mixed virus. So that all being said, we did a variety trial with a susceptible. We used Myrtle in this case, so higher is worse. And then these are all resistant varieties, claimed resistance. But as you can see, red snapper has similar virus level as the susceptible. Everything else had lower than the susceptible, and then Jolene and Grand Marshal had the best resistance. Grand Marshal, I would say, is the industry standard, so that's good, and this kind of validates our results. But also, we've seen red snapper fail in commercial fields as well. So as far as yield, Camaro, Grand Marshal, Jolene, and 2255 were the highest yielding. Camaro and Grand Marshal were better than Varsity. This one didn't have a problem with virus, but it just wasn't high yielding under conditions. This is red snapper, where I'd say intermediate resistant, and then the lowest yield was in the susceptible. So because of that, we're recommending the Grand Marshal, Camaro, and Jolene. The Jolene was early and had really nice fruit quality, and this is from a couple different companies, Cicada, as well as Bejo, so a little variability there. We don't recommend anything that's susceptible or Varsity because it didn't yield well, or red snapper because it actually didn't have that good of virus control. So this year, we were supposed to do a heat set trial, but of course, we went ahead and took virus ratings as well. And so this is a part of a grant that I'm involved with, and it's got Florida varieties, commercial varieties, and then Texas A&M. And what was excellent was the Texas A&M cultivars, really low. So again, you got your Grand Marshal, did well in the last one, but some of these are excellent, better than the industry standard, it appears. Again, for tomato chlorosis virus as well. I don't know if Amol's still in the room, but, well, I'll keep talking about this. I'll talk about his stuff later. But interestingly enough, or oddly enough, or typically of research, of course, this was a heat set, and we got frost. So we had yield ratings. We had a early November frost this year, and so these two that with Texas A&M had high yield ratings. We did a 1 to 9 rating. 9 was the best, but also we took bacteria ratings. They had the highest bacteria, so still needs to be worked out. Seems like maybe Texas has got better virus resistance, but being a dry climate, they don't have developed for bacteria, whereas it looks like the Florida, we might be able to bring better bacteria resistance. Now I'll give a shout out to the breeders. We're currently doing ongoing trials with watermelon squash with Dr. Cecilia McGregor. She's next door giving a talk. Like I said, the watermelon's looking excellent. Squash needs some work. With Amol Nankar, our vegetable breeder in Tifton, the tomatoes that I saw were better than a commercial standard out of the gate of the lines he's got. He's developing them. But Grand Marshal was about this high, whereas the other ones went to the top of the stake, looked green and pretty, nice fruit. So really, I'm really excited about the tomatoes that were being developed in Georgia under our conditions, not coming from California, Europe, etc. The snap beans, Bhavesh already talked about. Again, we know Caprice is the worst. You could pick that out in every plot. PV857 looks good. Momentum, Prevail. Syngenta has some good varieties. We've got HM Klaus that we're working with that I showed you looking good. So I think there's relief on the way, and we're continuing to work with these companies and do screenings in South Georgia. I just want to highlight the teamwork that everyone's doing a part of. One thing that I don't think anyone's presenting here is cover crops. Am I over? No. Keep going? Okay. I saw someone look at it. Watch. Interesting enough, with the sustainability specialist Taylor Singleton, they were doing cover crop work with between row grass cover crops, and they found much less whitefly and less virus in the cover crop. Philip, our entomologist, he's seen the same thing. There's less whitefly in weedy fields, so it just is a visual thing where it seems like they don't see the small plants and get on them early. Working with storming insecticides, repellents, as limonene, reflective mulches, etc. Next steps, we got some good early material, but after it goes to the breeder, I'd like to see it get good yield, quality, size evaluation so we can make good recommendations. Also, I want to focus on ethnic vegetables. Bitter gourd, chayote, other cucurbits that are same family but totally different genus and species from tropical origin, whether they're from South America, Africa, or Asia, that were from areas that might perform well to these more tropical viruses. And with that, I conclude my talk and answer any questions.
Any questions for Dr. Ted?
Dr. McAvoy,
Yes.
do you have a chance that we're going to have a public snap bean variety, or are you developing these lines to pass on to the commercial guys?
You'd have to talk to Amol. The good thing about snap beans, because actually jogged my memory, is it is a pure line. Everything else mainly is hybrid, but snap bean in particular is a pure line. I think the idea was to give it to companies. You could talk to Amol, and I'm sure he would fish out the royalties. But as far as the beans, I'll just say one more thing. They look good for virus. Of course, we got to work on lodging and pod color because you still have to spray because they were still getting bleached. But virus, they look good.
We got the capability of sending them out west, getting them produced. If you could develop a variety, a public variety, or a variety that we can buy from you instead of sending these commercial companies, it's save growers in Georgia a lot of money.
Well, I wholeheartedly agree. It's my opinion to release your own varieties. I know, but I'm not the one in charge of that. And I would just maybe pick his brain about the possibilities there and the support. I think basically bringing it all the way takes longer, and whereas if you kind of fish out a gene or some germplasm, then they take, it's like a handoff. But yeah, if you could convince them to bring it all the way, I wholeheartedly support it. I know that's what we've been doing. Georgia's been doing with cotton and peanut, and I think it would be a good thing. Yes, ma'am. Yes. I gotta go way back. Yeah, that's what it's Sarah Smith, H.M. Klaus, and Immokalee. I would refer to questions to her. They're not named varieties to my knowledge. But I don't know if they'll go commercial or when.
All right, thank you very much, Dr. Ted. We have our next speaker. Next speaker is Dr. Andre da Silva. He is the Associate Professor and Extension Vegetable Specialist in Auburn University. You can just plug it in.
Everybody's tired. Lunch was good. Everybody had that bun full of meat. Like me, probably want to take a nap in the hotel, but I will try to be brief. This is going to be like a continuation. Actually, I would say that my talk and Dr. Rodriguez, who's going to follow up, should be a little bit before Dr. Cremon's. We are a team in Auburn, and since we are in a growers meeting, I will not get into much detail on research. I want to transfer to you guys what we have been found in the past and what we are doing, as well as what Dr. Rodriguez is going to do. So our title is a little bit long, but it's like we want to show the strategy that we have been investigating in Auburn for whitefly control, mostly in tomatoes, but also talking about the drones, chemical application, and some of the genetic resistance that it's very similar to what some guys in Georgia is doing, but we are taking a different approach that is understand the plant resistance. We do a lot of variety trials, very similar to what Dr. McAvoy showed here, so I will not double dip information. You guys already have that information with him. Our results is pretty much similar, so I will try to go quickly. But just for you guys to understand the scenario that we are in Alabama right now, I want to talk about what we have been seeing in the past years. So in 2023, once we started like our tracking our whiteflies, we start to see most of our whiteflies in this region here in the southeast, where we border Alabama and where we border Georgia. What's happened is, as the summer comes, whitefly move north, and they enter in our state. So we start there. Pressure will report in the southeast. 2024, our pressure moved not only here, it's moved over here and southwest. So we saw that. Some scattered locations in central Alabama were seen. But this year in 2025, our pressure was lower, but still, I had growers reporting in the entire center to south Alabama. And the reason why it was discussed before is because we saw problems in vegetables, row crops, increased the reporting in greenhouses, like I was talking about earlier in the questions from Dr. Duda to Dr. Cremon, is most of our greenhouses are located here. But if you don't know, our nursery industry in Alabama is the largest in U.S., and they are located here. So we have a lot of report of whitefly coming from the nursery industry. So what they do is they winterize it. They stay during the winter in our nursery industry, which is the largest in the country. And then in the summer, they are coming to our vegetable crops there. And that's when we have problems. And now, herbs. Herbs is something that it's been a big problem for us as well. So we have a law that we are dealing. While I understand that Georgia, Florida, they have a large industry of vegetables. For us who are smaller in the vegetables, we are vast on the specialty crop, a lot of diversified growers. So our problems come from everywhere. So we need to deal with everything. So just keeping it in mind. So what we did this past year is we continue our drone base. For you guys who were here last year, I talked a lot about drones, how to do that. We continue to use drones to spray for whiteflies. We increased our trials. We are now investigating like where we should be applying more, in the top of the leaf, in the middle leaf, in the bottom of the leaf. We did trials where we evaluated different stage of tomatoes in past years. So we tried it in the early stage of development, we sprayed it. In the mid of in the reproductive stage of development, and in the fruit, and we spraying those crops. So this is just a record of what we have done. What we basically did is this year we reduced our treatments, and what we evaluated was a high quality final drop, where we evaluated our flight speed at 4 meters per second, our droplet density at 150, 250, and 350, and those are for the fine, medium, and coarse droplets. So we did 3 treatments. I don't want you guys to understand those parameters, but I want you to understand what is the size of our droplet and how we did apply. So we have a fine droplet, a medium droplet size, and a coarse droplet, where we call high quality fine droplet, high quality medium, and a high efficiency coarse droplet. So that's what we evaluated. And we did it at our first trellising, our second trellising, our third trellis. As you see different growth stages. So we want to see when we should control better whitefly with our drone treatments. What we found is that for our coverage, if you are spraying and want a good coverage of your canopy, you should be using our high quality fine and our high quality medium. That was where we have our best treatment. It's going to affect better the top of your canopy, and you should be doing that in your third application. It's going to be your best coverage. Of course, you have a higher canopy, you're going to have a better coverage. Early in the stage, you will not have that much coverage. But still, you see that always in the upper, it's our better application. That's for coverage. Droplet density is the number of droplets per square inches. In this case, we are showing in cubic in square centimeters. But again, we had our best treatments for whitefly control on the high quality fine and high quality medium. Actually, we are making recommendations for our growers, if you are spraying with drones, to contact insecticide to get a 50 to 70 drops per square centimeter, or if you have a systemic one, 20 to 30 drops per square centimeters. Thinking about 1 inch is 2.5 centimeters. So that's just for you to have an idea. And then the application on the treatment, like we always have those treatments as the best, high quality fine and high quality medium. So for us, what we identified that it's for whitefly, you should try a smaller droplet size, the fine and the medium. Coarse was not something that we are looking for. So that's what we are recommending right now. This is just the upper or the bottom canopy. We already expected that as higher is the plant, the top of the plant will receive more insecticide than the bottom. So we did the same thing for the deposition, is where it's being located. Again, we will consistently have the high quality and the medium droplet a better response. We have the same thing for the application times and also in the upper of this. So what is the current recommendations? We are making recommendations to growers. What we are saying is they should try lower speed, higher application volume, and fine droplet size to increase their coverage, drop density, and deposition. This will increase timer over target, great liquid delivery, and denser spray cloud. Coverage, droplet density, and deposition are consistently higher in the upper canopy, so try to focus on the upper canopy to spray for whitefly. That's what we are doing. And spray early in the season, you increase your chance to reduce the lower canopy. So what we are saying is, like if you are using your boom to spray the whitefly, to spray for whitefly in the early season, go with drones, because you can go faster there and you can target the whitefly. Because if you have whitefly pressure early season, you're going to have problems. Yes, we have virus, like we were discussing early. We're going to have virus mainly in tomatoes. That's going to be a big problem. So early in the season, the use of drones, it's going to be the future for managing whitefly on spraying programs. I'm not saying only for is the only solution or the only management, but it should be part of the IPM program. When we try some insecticide, we had the same thing that was before in the last 2 years. We have tried those ones. We have a better results for X-Rio and Savanto rotation is still our best target that have the lowest pressure, and the RM1963, which is a biological one, we still also have some, the best control. It was very similar to our X-Rio and Savanto. It's our best recommendation currently, and it allows our best yield because reduce our pressure. Although this one had no significant difference from the other ones that had no significant difference, who had the lower yields. Yes.
What type of biocontrol is that?
Paulo, do you remember exactly? I have that one, but it's a product that it's supposed to be in the market like this year. We received it from a company from California, and I can give you the information later, Ted, no problem at all. And those are basically our, like I said, X-Rio and Savanto is still our best recommendation. NEC for nymphs. This is for control of adults. With this, I would like to invite Dr. Rodrigues to talk a little bit about what we are doing as far as understand the plant resistance on whitefly, and she's going to give you a better update on that.
What was your height of the drone plant?
We keep it 3 meters above the camp all the time that we are spraying. So if plants are small, 3 meters, which is 3 meters is 10 feet. So if you're doing like, if plants are taller, 3 meters. And then, but when you trellis it, it must be 3 meters from the stems. Otherwise, it's going to fall on the crop, and it's happening in a grower's field.
Right. And is it better than the regular boom spray?
Yeah, it's more efficient.
Okay.
It's efficiency.
Control is.
Yeah, you reduce the water requirement, you reduce the product use, and you increase the speed and labor. But we did not do economic analysis yet. So don't ask it right now.
Gotcha. Thank you.
All right, so for this part of the project, we're trying to understand some plant traits and natural defenses in the plants that are interconnected with attractiveness or repellency of whiteflies into the plants. So just for you understand, tomato plants, you know, when you get a leaf of the tomatoes, you see those hair-like structures on the bottom of the plants. We call them trichomes. And those trichomes, they have different types of trichomes. You cannot see them with bare eyes, but when you look at the microscope, there are 2 major different groups, glandular and non-glandular. So I'm going to show a microscopic picture here so you can see better. With the glandular trichomes, they literally have a gland on the end of their tip of their hair, and those glands, they produce chemical that they help the plants fight against predators and all of them. So those chemicals we call secondary metabolites. A lot of them, they are volatile compounds. So in this case, we're studying this because over the years, the breeding programs and the development of new varieties, those types of trichomes, they shifted from mostly glandular to non-glandular. So we're looking at what is the relationship with these glandular trichomes and the metabolites that they produce, if they play a role on an insect repellency or attractiveness. So yes, as I mentioned, we are trying to identify what mechanisms or plant mechanisms of resistancy and the sweet potato whiteflies in tomatoes. So first we categorize these trichomes into different tomato plants, and we also verified the volatile compounds, the profile of these compounds produced by those specific plants that we planted over these years. So we ran 2-year trials. So the first year we got we tested 4 commercial varieties and 8 wild accessions. So those are the wild tomatoes. We obtained them from the USDA repository. And for this year 2, we tested some of the same varieties, but we added some different commercial. So we tested 5 commercial varieties and 7 wild accessions as well. So this was all done at the Wiregrass Research Station. So right at Dr. Da Silva mentioned where there was a big pressure around Dalton area. So that's where the experiment was conducted. So briefly, just to show you, based on the accessions and varieties that we tested, we counted eggs, nymphs, and adult whiteflies. So looking at the overall results, we could see a trend in terms of egg deposition, nymphs, and adults. So in general, we could see in the commercial varieties at the bottom here, you see that there was some varieties, some plants, they have more deposition, egg deposition, and then also some adults. Although with this specifically 3 wild varieties, wild sessions, we saw a reduction in eggs, a slight reduction in adults compared to the other ones, but a very lower concentration of nymphs. So I think Paulo can follow up on me that they were depositing the eggs, but the eggs were pretty much not hatching. So something on these 3 specific plants that they had traits that we were looking into investigating what was going on. So that's why we come into this characterizing the trichomes. So interesting, we saw that most of the plants, especially the commercial varieties, they had no glandular trichomes or very minimal glandular trichomes. While the wild accessions, they have a larger number of glandular, and specifically these 3 ones, again, they have a higher concentration of glandular trichomes. Remember, the glandulars are the ones that are producing most of those secondary metabolites. So here's just a picture so you can visualize, that's very clear, those glandular trichomes compared to the commercial varieties. They are very, very hair-like structures, so they are non-glandulars. When we look at the chemical profiles, again, amongst those 3 wild accessions, 1 came out with a very unique and specific chemical metabolite profile. So giving us that this relative abundance of terpenes. Terpenes is a class of the metabolites that we are looking into. In these wild tomato sessions, they were potentially play a role on repellency for whitefly on those plants. Or maybe that's the follow-up that we are working with Paulo and doing like target laboratory target and preference testing with this specific metabolites and insects. So in general, for this specific project that we are doing, we found that this, in case this wild accession, it has a very significant resistance to whiteflies compared to the other species. And we found I have very high density of glandular trichomes on this accession. And then the specific class of metabolites that was correlated and it was present on this plant. So that's the next step for us, is selecting more varieties, more of those same species, but also extrapolating to other different species. And also, I think part of that, what Paulo presented, the greenhouse project, those all of those trials that he presented, we collected samples that we are analyzing those the chemical metabolites, so we are correlating with the information that he found in the greenhouse. And we're also working, we have a herbs trial that is probably going to start in a few months. That we're also going to be looking at traits, metabolite traits, and then into these plants as well. And we are trying to understand how these secondary metabolites play a role in insect physiology. And the ultimate goal is to maybe work with the breeding programs and try to bring back some of those traits into new varieties, so we can work as a new strategy to improve resistance on those plants.
One of the things that we are doing here is not only for the trying to give growers the recommendations, like we are trying to do real time and next year application, like the use of drones, variety that Ted is doing, similar to what Dr. McAvoy is doing, variety trials, insecticides trials. But at the same time, we identify what are the metabolites that create resistance in plants. If those are the ones that create resistance in tomatoes, but now we are seeing the internalization of the whiteflies in the greenhouse on herbs. So what's happening on herbs? For example, we see that the infestation in rosemary is being reported as high, and that's when this company is growing their transplants for rosemary. So it's full of whitefly, the rosemary greenhouse. The other one is sage as well. It's another one that is very susceptible.
And mustard.
And mustard. They are the ones that we are seeing a lot of whitefly. So what are the metabolites that those crops have? And because when you're talking about trichomes, if you compare cotton with tomatoes, whiteflies prefer hairy cottons. In tomatoes, whitefly don't prefer hairy tomatoes. So that's what we understand now. Why hairy tomatoes and why hairy cotton, why not hairy tomatoes? We know that now. But what about their herbs? We don't have hairy herbs, but we do have like the metabolites produced by those plants, and that's what we are trying to understand. So what we are trying to do is solve immediate problems for the development of IPMs, but at the long term, what we're gonna be doing is provide the companies information for breeding program, but also chemical programs. If she come back one of the slides, we already know what are the metabolites in tomatoes. So we know one more. There we go. We know that those are the metabolites or all the chemical profiles that require. So they can develop like herbicides or all those ingredients that can be used for spraying on tomatoes.
So if I understood correctly,
Yes.
trichome glands are the ones that provide all these metabolites. So when creating new varieties of, in this case, tomatoes, are you looking to increase the glands, the trichome glands, to improve that without having the farmer to buy those metabolites that will help?
Yeah, we don't want farmers buying the metabolites. What we would like to is provide the information to breeding programs. So because those are wild tomatoes, if you taste them, it's not good. So during the breeding program over the years, what's happened is they increased the flavor of the tomatoes, but they lost the protection that they used to have. So now we are in the opposite direction. We are trying to increase protection without losing the flavor. So what they do is breeding programs need to introduce glandulars, trichomes again, in order to maintain the protection, but also keep the flavor.
Yeah, we're shooting for maybe increasing those natural defensive mechanisms that the plants used to have it, and now with new varieties and breeding programs, they focus in one part, which is quality, taste, and all of that, and then a lot of traits have been lost over the years. So that's what we are trying to understand here, and then hopefully the breeding programs will start focus, maybe incorporating those things in the new varieties.
All right, we gotta move on. We got last two speakers. Thank you very much, Dr. Silva, and everyone. Second last speaker of the day and of this conference, I think. So let's welcome Dr. Alvin Simmons. He's the research entomologist at USDA, and he's going to present some of his research today. And I'm pretty sure Dr. Simmons will be on time and will wrap up in the next 15-20 minutes.
Of course, I'll be on time. And because the past two speakers were a team speaking, there will be a team speaking this time, but with props. Yeah. So the USDA vegetable research in-house several different locations: Arizona, California, Florida, Georgia and South Carolina, and we'll be very briefly speaking about a little bit about research, in-house research projects at these locations. In Arizona, well, first of all, these are projects which are either in-house virus projects or entomology projects. In all of the entomology projects, we just got recertified or being recertified for a new 5-year plan. And for the virus projects, well, they will be in about a year to be modified. In Maricopa, Arizona, their focus has been on biocontrol using thresholds for whitefly IPM, especially in cotton and milling. In cotton, because of the BT, they are looking at the non-target impact, which primarily with activity on ligas and thrips. In Salinas, California, research being led by Bill Wintermantel, in which they have developed 2 methods of identifying viruses as a single infection or as a mixed infection. And this also allows to look for the level of infections in both the insects and also in the host plants. And this also, they're using this method to look at new and emerging viruses to be able to identify those. And because, as we've heard over and over again, viruses is a big problem of the whitefly problem. And not only in the southeast, but also in the southwest of the U.S. When it comes to the whiteflies in the desert southwest, it's so easy for a population to really build up, especially in milling crops. And then they move over to rather, between the milling and the cotton, in which they are seeing a combination of single and mixed infestation, as illustrated by these yellowing in the pictures here, and it can affect the entire field. A problem in which has been detected initially in 2023 is the watermelon carotid stunt virus. This is a problem that's initially in Mexico detected, and then soon thereafter in Arizona and in California. On the one hand, we are fortunate that it has not yet, and again, the word is yet, spread beyond those areas. However, the intensity has increased. There are a lot of wild hosts for the virus that are being detected in those areas. So when I say not yet, it's just a matter of time for, that's what viruses, of course, do. They spread. Movement of infected plants can facilitate the spread, and of course, movement of the whiteflies themselves when they're infected can facilitate the spread. In early last month, the Emerging Virus and Cucurbit Working Group published a fact sheet online. And there are lots of great information on that fact sheet. Also, there's a video. And earlier this week, they published a PDF of this. And again, lots of great information there. And they do plan to, the group does plan to do some webinars later this spring to help let growers and extension have more information about this whitefly transmitted virus. In Florida, in Fort Pierce, there are two groups, one entomology, other virus, of working with whiteflies and whitefly transmitted viruses. When it comes to whiteflies in Florida, it's almost every year there's a new invasive whitefly. It's just like, it just happens. And so continually having more viruses that are invading the U.S. in general, but also the whiteflies, new species popping up. And Dr. Sandy McKenzie on the entomology group is leading, and that's including by the use of Banker plant system in which papaya, using the papaya whitefly as a host for biocontrol parasitoids. And these whiteflies do not feed on poinsettia, the target crop, or on tomato, another target crop. And this is a system that more conducive of working in a enclosed environments such as a greenhouse. Also in Florida, in Gainesville, whitefly work is being done on the biological-based taxes. And I'd like to pause to make one note that at the ARS research on whiteflies has been negatively impacted because of the loss of some of the researchers during this past year. So there are a few vacancies until we get those refilled. We're continuing pushing forward with the research, including collaboration with other research outside of USDA ARS. In Georgia, Tifton, Georgia has the project on pests in the landscape. Because whiteflies, they are in the agricultural landscape for sure. For Tifton, the entomologist that was leading the research on whiteflies there has moved on. So currently there is a vacancy there. Also in Georgia, in Byron, Georgia, at the Fruit Tree Nut Research Laboratory, David Shapiro Elan is entomologist leading collaborative work with University of Georgia with the entomopathogens. And when it comes to entomopathogens, always been, when challenged, been the sunlight or the moisture. And research in which Jermaine Pierre, oh, he's right there, before he moved, was part of the team and led research in which, on one hand, we see that by having pheromones treated entomopathogen nematodes, we can enhance the efficacy, but by adding the gel factor, a dramatic increase in the efficacy. In South Carolina, there are 3 projects concerning whiteflies. 2 are led by plant pathologists, Kashu virologist for one, the other Chaker Kusit. and also the entomology being led by me. The whiteflies, as we've already heard, feed on a lot of different plant species, over 1,000 different plant species. We are continually and presently looking at different potential plant hosts. And although I was hoping to share some of the results from some that we've just been looking at this past summer and fall, but with the government shutdown, well, things got a bit disrupted there. But I would note that there are several unreported species that we're currently looking at that we will see that they can and will feed on, and I'll be sharing that later. But the beauty and the sad thing about whiteflies on different crops, some crops, some plants hosts, they are great in their performance. Others, fortunately, they are very low incidence of them. Even though they may be able to survive on these and develop on these, the downside is that of any of these which they can survive and develop on could be a potential source of a virus if that host has a virus that it can transmit. And speaking of virus, in sweet potato, there are several viruses which are transmitted by insects, notably by aphids, but especially by whiteflies. In Biguma virus, there are at least 11 known ones that are transmitted by Bemisia tabaci. And among those is the sweet potato leaf curl virus, one which can be devastating to the crop. And we know that a close relative of the sweet potato is morning glory. There are lots of species of morning glories. And we've seen all of these species can host the whitefly and, unfortunately, this virus. In the field, the sweet potato leaf curl virus can dramatically reduce the yield in sweet potato. Some cultivars are less impacted, being more tolerant, than others, as illustrated here. We did an extensive wild-scale experiment, a grow-out, and we do not see the sweet potato leaf curl virus as being seed transmitted. A colleague, Dr. Sharon Andreessen, one of currently the only other entomologist permanent in Charleston, which is doing research on transmission of whitefly viruses. And that's including looking at the role of symbionts, and also integrating the factor of natural enemies in the complex of the virus, plants, insect interaction. The amount use pesticide program through the IR4 program, which is part of a national effort to help provide options for growers because the pesticide industry, their incentive is decreased because of less return. And so this national program helped provide options to register for growers. And for the research in which ARS does in conjunction with the National IR4 program. It includes all classes of pesticides, including pesticides which impact whiteflies both in the field and on greenhouses. The effort by ARS results in about $1.3 billion each year for this overall program itself. In acknowledgement, just a general acknowledgement for the ARS researchers in ARS in Arizona, California, Florida, Georgia, South Carolina, and the whitefly team, which including collaborators, ARS, University of Georgia, Fort Valley State University, Auburn University, and other collaborators. And according to my time, I have 4 more minutes, so there should be time for questions.
Thank you very much, Dr. Simmons. Any questions for him? That was great, on time.
Yes.
The invasive every year in Florida, whiteflies seem to come into Florida every year. Is there a general route? Is it coming in on plants, weather bringing them, or what's the route?
And if I heard correct, you did say Florida, is that correct?
Yeah, when you mentioned every year something, a new invasive whitefly comes in.
You can repeat the question for the recording, if you don't mind.
Okay. The question was, if I heard correctly, is of the invasive whiteflies that comes into Florida about every year. And the last part of the question was, what's the general route? It is mixed. One, it comes in on plants. That's one. Two, we know we always have weather systems, as in hurricane, and we have South America, we have the Caribbean. And yes, whiteflies can survive in weather systems and can be carried, like other insects can be too. Also, unfortunately, they do come in on airplanes, on cargo, again, on plants.
Following the same question, the weather systems, do they survive as an adult or egg or any, all of them?
Okay, for weather system, that's for the adults, that they can survive.
And for plants, it's like eggs or...
The plants, it's most probably the adults are easy to be seen, and they probably, if any plants are infected, they're probably going to get seen and get captured through quarantine, probably, hopefully. However, eggs and nymphs, they are less likely to be observed in those scenarios. And again, it's on the leaves, lower surface primarily, where they can slip by.
All right. Thank you very much, Dr. Simmons.
Thank you.
We'll go with the last speaker of this educational session. Our last speaker is Dr. Babu Srinivasan. He is the professor of entomology at University of Georgia. Please welcome Dr. Babu.
All right, thank you. I'm always seem to be a magnet for these just before beer talks, you know. Anyways, I won't try to keep you for too long. And what I'm going to talk about today is just vignettes of what you already heard, snippets, and just take you down the memory line as to how we got here and what we did in the last 10 years or so. More so in the last 7 years since we got the USDA collaboration with Dr. Simmons. And so that's the bulk of it, and some of us have been working on this before that as well. And then a few words of wisdom to conclude as to what we could, as to what the path forward is going to be. All right, so again, I'm not going to harp on this because you already heard about it. You have whiteflies, they are present year round, but for us, the problem is mostly in the fall, so that's when we have heavy whitefly pressure, and I'm going to tell you why that's the case as well as we go forward. Okay, so you heard about different viruses, but I'm also, since I'm an entomologist, I'm going to give you my 2 cents on whiteflies as well. They're not all the same, so we have a problem in the last 10 years or 15 years, as I said, because of the viruses they transmit. But also, you know, we didn't have this problem before. We had whiteflies all along. So why is that we didn't have a problem? That's because we have what's called as invasive whiteflies. Okay, those came from Middle East as well as from the Mediterranean area. So the one that came from the Middle East is called, from the Middle East Asia Minor region, is called as the cryptic species B. For those of you not familiar with this, whiteflies are cryptic species members, and depending on however you characterize them, there are several different cryptic species, okay? Some would say 40, some would say 6, and so forth. But this is probably the most comprehensive work that was actually done by Dr. Judy Brown from Arizona, and she's used biogeography as well to substantiate her findings. But the bottom line is we got hit with biotype B, or cryptic species B, and that's when all this problem started. And why did it start? Because the cryptic species B is a very efficient virus transmitter. And Q as well. It was introduced later from the Mediterranean area. But luckily, so far, Q has just been mostly restricted to greenhouses. All right? So whatever you find in the field is B, and B replaced what is known as cryptic species A, and A refers to Arizona. So that's what we had, and that got displaced by this invasive whitefly that we got from Asia. All right? That's when all the problems started. All right, so again, this is just to show how homogeneous the whitefly population is, okay? So we sampled over different counties, and I'm going to make the long story short. So we have different ways of molecular tools to characterize what cryptic species it belongs to. And again, as I said, B is the predominant one. We also found 1 or 2 locations where Q is present as well. That's in North Georgia, mostly restricted to greenhouses. But whatever you see where the crops are grown, they're all B. Okay? And we also sampled them from different crop species, and this is what we call as principal component analysis. But the gist here is that these are samples from different crops and different farmscapes. And what you see is no clustering. And what that means is basically all these whiteflies are one and the same. Okay? And so this is a problem for us because we grow over 1 million acres of cotton, and we grow a few thousand acres of vegetables, right? And you have this influx of cotton when they start senescing and defoliating, and then they move to vegetables, and the vegetables are basically sitting ducks, right? And again, the main issue is it leads to colonization, and colonization leads to population explosion. And there are some issues with feeding the associated with whitefly known as irregular ripening in tomato, and then you also have the silvering that the others talked about earlier. These are problems, but these can be dealt with if you use insecticides, and you can deal with them. But the main problem is that you can actually effectively still deal with is transmission of viruses, right? So let's see. Okay, whiteflies transmit viruses. You've heard this story throughout today as well. So I'm going to tell you, just tell you there are different ways by which whiteflies transmit these viruses. I'm not going to go into the details. It's not necessary. But also that why are whiteflies efficient transmitters of plant viruses, right? And the bottom line is whiteflies feed on the phloem, which is located centrally in the plant. And what happens is that when they feed on the plant, they make very minute punctures, and then they cause minimal cell damage. And for those of you not familiar, viruses are obligate parasites, which means they ought to have live cells for the viruses to multiply, right? So whiteflies cause minimal cellular damage and then seal the plants when they withdraw their stylets, okay, after they inoculate the virus. And that's why they're able to transmit the viruses very efficiently, right? And you've heard about all these viruses. The only one you probably didn't hear about as much is the squash vein yellowing virus. And these are the RNA viruses, and this one's the number of DNA viruses as well. So for the sake of convenience, I categorize them into different groups. One is chronic viruses that have come have been introduced into Georgia and Florida or in the southeast, and then they've decided to stay. And some recent introductions, we don't know if they're going to stick around. And then the ones that are on the watch list that have been found elsewhere in the U.S. or in the area in South America, so forth, and have potential to be here sometime soon. Okay, so I'm going to run through some of those really quickly. So you heard about this, tomato yellow leaf curl virus. That's probably what started it. It came towards the end of the 90s, late 90s, early 2000s, and it came from the Caribbean islands, and they have been here ever since. And this one's tomato chlorosis virus, which Ted talked about as well. We found this in 2011, and it's been there since then. And often tomato chlorosis virus and TYLCV are transmitted together. Well, even though one is an RNA virus and it's transmitted semi-persistently, and the other one's a DNA virus, but they're all transmitted together by whiteflies because they're all present in the phloem. So all the viruses that infect vegetable crops are all found in the phloem, and whiteflies are phloem feeders. That's why they can transmit multiple viruses, right? Now this one's another one, cucurbit leaf crumple virus. You've heard about it. It's a major problem in squash and some cucumbers and so forth, mostly squash. Yellow stunting disorder virus, that's also a chronic virus. This came here about, I would say, 2015, 16, and we've seen it every year. And the whitefly is the gift that keeps on giving, so we keep finding newer ones. This one's squash vein yellowing virus, mostly a problem on watermelon and Florida, not so much, even though it's been detected in Georgia and Florida, not so much of a problem for us because it's in the fall, we don't really see a whole lot of watermelons that are grown in Georgia. Vine decline, yes. So basically what it is, it kills the entire plant. And that's the virologist from Fort Pierce, Scott Atkins, that Dr. Simmons just talked about. All right, so again, these are some of the newer ones. This was my grad student was doing some work, and then he found another new virus. This is the cedar golden mosaic virus. Mostly a problem on snap bean, which Babesh talked about extensively, but it's also got an extensive host range that includes a number of Malvaceae hosts, such as okra and hollyhocks and so forth. Also, it infects tobacco. So this is mostly a problem for beans. And this one was what Sudeep, our virologist, found a few years ago, cucurbit chlorotic yellows. These have been in other places in the U.S. before, but have been recently introduced into the Southeast. So those are some recent introductions, and Dr. Simmons just talked about this, watermelon chlorotic stunt virus. It's been noticed in California, probably in Arizona. Mostly on the West Coast, not so much here yet, but we even sequenced some samples this fall, but we didn't find it. So another one, this is actually not whitefly transmitted, but it's tomato brown rugose fruit virus. It's a major concern because this is a tobacco virus, and this can shut down greenhouse seedling transplant production. But we have been looking for it as a sort of monitoring effort, and we haven't really come across that in Georgia yet. Another similar one, another tobacco virus, is the cucurbit green mottled mosaic virus. Again, there's been vague reports of one or two places, but not really so much of a problem yet. But these are causes of concern, and we should be on the lookout for. So that's just to give you an idea. Every year, like I said, the gift that keeps on giving. And this is where we are with the number of viruses that we have. And as I said before, these have not just single hosts. They have multiple hosts, what we call as alternate hosts. As a consequence, they've established themselves in the landscape and in the production system. And mainly because there are alternate hosts, and the alternate hosts could be crops themselves, as in that case with cucurbit leaf crumple virus, and in the case of tomato yellow leaf curl virus as well, or it could be weeds like amaranth and many other species that we have in the landscape. So yeah, there are plenty of hosts, so not only are these whiteflies polyphagous, and the viruses themselves have a broad host range in many cases. And then once they're here, and the weather permitting, they're good to stay. So yeah, here are some more examples of other viruses establishing themselves in the system. And again, there's plenty of work that's been done over the years that documents these and how these hosts themselves can serve as reservoirs for either the virus or the whiteflies or both. And when I talk about whiteflies, I'm just talking about this one particular species and one particular cryptic species. So again, as to why that is important, as I said before, there are different cryptic species of whitefly, and when I say cryptic species, and what it means is that they're morphologically indistinguishable. Genetically, they are different, but morphologically, they're one and the same. So that's what we have here, B and Q, and we have a number of these new viruses. And what we found over the years is that the cryptic species B alone can efficiently transmit virus. Okay, this B can transmit both TYLCV leaf crumple virus as well as cedar golden mosaic virus. This one's an old world virus. That's what came here first. And what we are now seeing is more new world viruses coming in from South America. Okay, so these are bipartite viruses, at least most of them are. And what you see over here is that the way these cryptic species interact with the virus is fundamentally different. And what you see here is the gene expression in whiteflies when they acquire one of these viruses, right? And you can clearly see, sorry about that. You can clearly see how different the whitefly responds to, differently the whitefly responds to each one of these viruses. So we, in our lab, have spent the last 10 years trying to characterize a lot of these interactions. The bottom line is they may look the same, but they're not the same, right? So it's a very complicated process. All right, so and then we also heard about mixed infection, and I'm going to tell you why this is important from a breeding standpoint as well as we get through these slides. So what you have here is a squash crop that's infected with C. DV, leaf crumple, and look at the phenotype of these plants when compared with non-infected plant. And then you have one that's infected with two viruses, right? So this is like getting COVID and flu together or flu and a norovirus together, right? It's just not pleasant, and it's the same for plant. And this is important just because you have resistance. When you have resistance against one virus, and when you're infected with multiple viruses, then the resistance that you have for one can get broken down, right? That's also what's a problem for breeders, is because there are resistance sources, different resistance sources for different viruses, and to introgress all of those takes a lot of time and effort. And even if you do that and you get a new virus, that can be easily broken as well. So again, that's just shows you the height difference when they get infected with two viruses and then one virus as well as the control, which is not infected with any virus. All right, so again, yeah, go ahead, please. Yeah. That is correct.
So that means that all the efforts that the breeder did for the A to control A virus are not going to be effective in the future because it already has.
Yes, that is correct. So what happens is when you have resistance, for example, in the case of, I'll talk about this as well, when you have resistance against tomato yellow leaf curl virus, right? This resistance is specifically for that virus. It's basically a gene for gene resistance. Okay, so TY genes confer resistance to TYLCV, right? But then when you have TOC, which is tomato chlorosis virus, right, then that resistance actually can be subdued or even broken, all right? So yeah, that's the problem. Yeah. Okay, so why is this a problem? I know you had a lot of talk about managing whiteflies with insecticides, but when it comes to dealing with virus transmission, the threshold for whitefly is just one. If you have one, a single insect can transmit a virus to a plant. Just to give you a magnitude of the issue, you may have in the fall up to 150, 200 whiteflies on a single plant. And then you have millions that come in from cotton, or billions rather. And then you have 3,000 tomato plants or 4,000 tomato plants in an acre. It's just nothing. They're sitting dogs, right? So it's impossible to manage the situation without addressing the virus, right? Okay. So host resistance is the number one option. It's been proven time and again in different places across the world. If you have host resistance, you're good, but it's hard to get it for most of the viruses that you have because it also takes time to develop resistance, right? So So what do you do in the meantime is you try to basically do a Band-Aid approach with cultural tactics and chemical tactics and so forth. Yeah, so again, this shows the difference between a resistant variety and a susceptible variety. With tomato yellow leaf curl virus, which was what, 10, 15 years ago, I took this picture. And what I want to show here is that resistance is not harmless. Resistance is not going to sustain forever. What you have is these gene sequences of the viruses collected from susceptible plants at the top and resistant plants at the bottom. What you see here are multiple nucleotide changes, right? Which means the virus is producing a selection pressure on the plant continuously. We still don't see evidence for it breaking down, but that's a possibility because a number of these changes actually could break down resistance, okay? And those are concerns which Ted talked about, has been proven in a couple of places at least, one from Georgia and one from Spain. So if you have more than one virus, you might lose the resistance that you have for the other virus. And this is another example that I've been working with Babesh, and he talked about cedar golden mosaic virus as well as cucurbit leaf crumple virus. It's easy, there's more lines that show resistance to leaf crumple, but not so much to cedar golden mosaic virus. But then when you have both these viruses together, you can see the plant that's in the middle that's infected with two viruses. And it's just not going to hold up, right? All right, so squash, I'm not going to talk too much about it because there's been already talks about it. You have zucchini that's a little bit more tolerant to the squash virus complex, but not the yellow squash. It's very susceptible. That's all a lot of work that's already been done and shared with you. So the bottom line is Cucurbita pepo has very little resistance to the complex of cucurbit viruses. So what happens, what could be done is to incorporate or introgress resistance from other wild species, and that's what the cucurbit breeder, Dr. Cecilia McGregor, is working on, and we've been helping her with screening some wild species. So basically is to incorporate resistance from wild species into cultivated pepo, and that often comes with the yield drag as well as the other important traits that you wish to have for the crop to persist may not actually exist. So there's going to be a lot of trade-off there as well. So here's again differences in whitefly numbers as well as virus severity depending on whichever wild species you use. Another thing that you heard a lot, people talk about the trichomes, and these are type 4 trichomes which produce, in this case, this was work that we did with people from Cornell, and they produce what you call as acyl sugars, right? And these are good when it comes to repelling whiteflies and when it comes to managing the whitefly populations, but not really nowhere close to good when it comes to managing viruses, right? Here's an example. These are acyl sugars, are nothing but fatty acids, short or long-chain fatty acids with sugar moieties. And what you have here is differences in terms of preference between a conventional cultivar as well as the lines that have acyl sugars. And you can see there is reduced preference in feeding, but when it comes to virus transmission, really not much of a difference, right? Every single plant is eventually going to get infected because acyl sugars help maintain or help reduce whitefly populations, but they don't prevent virus inoculation. So that's again the problem with having to deal with different modes of resistance. So the resistance is better when it's against the virus as opposed to the insect. So other ways that you can manage whitefly viruses is to manage if you're using seedling transplants, make sure that they're not getting infected in the greenhouse, use netting, use different insecticides or immune defense modulators that can help reduce virus incidence. And you heard about this, I'm not gonna skip over this. Reflective mulch is better than many of the insecticides that we've used over time. And insecticides, again, here show that they can reduce feeding, but not completely. So as a result, they will still find the phloem and inoculate the virus. And again, different insecticides. None of those actually work except for using row covers in the early season. And you take them off, especially during flowering. And insecticides are great at reducing populations of whitefly, but not really reducing virus itself. And again, you use throw the kitchen sink, throw different tactics together with the row cover, with the silver mulch, you may be able to make a better crop. But if you don't, it's sort of what you're going to end up with, right? Again, this shows that row cover is better. So again, just like what Bhabesh talked about, using a risk index, this is something that we developed for tomato yellow leaf curl years ago. So there are tools available which tells you which tactics you could use to mitigate the risk that you would have over the season. And as a consequence, you want to end up yourself in the low risk category and then make a better crop. So there are tools available for that, but again, the main driver of these indices are resistant varieties themselves, right? And I'm going to, so about a minute or so left, so I'm going to talk about where we should head from here on. This is sort of philosophical, but again, this is where the world is heading to, and maybe we can catch up and do better at developing resistance. Would be using molecular biology and molecular tools, which Anup talked about in terms of SNPs and molecular markers. Use CRISPR and AI. AI can help speed up the phenotyping process, which usually is the longest when it comes to developing a variety. So that can be hastened with using AI as well. And in our lab, we're developing methods to, or rather understanding the mechanisms of virus transmission itself. There are proteins of whitefly that interact with virus capsids or virus proteins, and then we're using that to suppress viruses. And eventually, we have identified a few compounds already. Eventually, we're going to try and use a nanocarrier material to see if we can exogenously apply insecticides, but these would be aimed at preventing virus transmission rather than targeting the insect itself, right? So that's that, and then there's a number of other ways that one could use to automate monitoring and sampling. This is where AI could come in. And this is an interesting instrument for greenhouse production. This is in Japan, and basically what this robo does is suck up all the whiteflies in the greenhouse. And so, like a vacuum, yes. So, and then another interesting approach is my colleagues in Italy actually use it for spittlebugs, and what they use is vibrations that the insects produce. They have characterized those vibrations, and then they're able to transmit these vibrations to the entire field and then repel insects from settling on those plants. So these are some cool ideas that are, taking shape and then could be developed and molded into down the road. So yeah, that's pretty much what I had to talk about, and I'm sorry I went over a couple of minutes. And so I want to acknowledge all the collaborators, USDA and the university collaborators from either from UGA or from elsewhere, in the funding that we have, agreement that we have with Dr. Simmons at Charleston with USDA, as well as the Georgia Commodity Commission for Vegetables. So I'll stop there, and if you have any questions, I'll be happy to answer them.
Any questions for? I have.
Go ahead,
So,
Vivek.
not all viruses have same severity, right? There are some virus diseases that are a little more yield reducing, yield limiting. Can you expand on that?
Sure. So your question is not all the viruses produce the same effects on the plants? Yes. So there are different species of viruses. It's like very similar to what your flu virus strains are, or your COVID or whatever, right? Some of us get affected more and some don't. So it depends on the plant's susceptibility to the virus. The same cucurbit leaf crumple virus affects squash, yellow squash, zucchini squash, watermelon, cucumbers. Each of these species would show some symptoms, but yellow squash is the one that gets affected the most. It's just the way the plants are susceptible to, the magnitude of susceptibility.
And it can go from 100% yield loss.
So if I were to remember these years correctly, it was 2016 and 2017. That's when this whole thing started, and I would say the bottom fell apart. We had leaf crumple show up for the first time. We had detected it before in snap bean, but not until 2016-17 in squash. And that fall, there were a number of fields, especially in Dougherty County, that were plowed upon. So yeah, it can be a problem. It can be a serious problem if you don't manage it. And oftentimes it's not a great idea to either grow pumpkin or yellow squash in the fall, but we do grow them because they produce more money, yeah. So, hey, go ahead, Jimmy.
So it looks like we're heading towards a lot of resistance breaking in the future. Do you know if they're working on any new lines that might help with that?
It's a whack-a-mole process, right? You develop something and it breaks. For example, it also depends on the kind of resistance. Tomato spotted wilt virus resistance, right? That's a dominant gene resistance. That's already been broken in tomato and pepper in many different places. These, luckily for TY, these are semi-dominant genes, and they don't offer complete resistance. The plant, you don't see any hypersensitive response or anything like that. The plants would still get infected, but they'll still produce yield. So there, the selection pressure is less, right? So in those instances, the varieties seem to have a longer lifespan, or shelf life, rather, but not so much in the case of genes that are, where you have gene-for-gene resistance. So, yeah.
All right. Thank you very much.
Not a problem.
And I have a token of appreciation for you.
Oh, thank you.
There you go. Yes, sir. Thank you very much. That concludes our vegetable educational session for this conference. If you have not answered the survey, I would like to encourage you that. And thank you very much for attending. We very much appreciate you all. Thank you. You all have a safe trip home.